Hi everyone, thank you for attending this session. I'm Sophia, I'm a first year PhD student at the UCL Cancer Institute, and today I'm going to present you my work. So we developed JIGSAW, which is the first method to accurately infer exact copy number from targeted single-cell DNA sequencing. So first, cancer can be seen as an evolutionary process during which cancer cells acquire different type of genetic alterations that leads to the formation of heterogeneous tumors that are composed of a subpopulation of cells, also called tumor clones, that are represented here in different colors. Among all the different type of genetic alterations that those cells can acquire, the most two frequent one are SNVs for single nucleotide variants here, and copy number alterations. The latter leads to either the amplification or the deletion of larger genomic region. However, those SNVs and copy number do not act independently of the genome, but actually they can co-occur on the same genomic region. And one of the many mechanisms that involve both SNVs and copy number are double hit event. So during a double hit event, one allele of a tumor suppressor gene will be mutated using a loss of function mutation to inactivate it, and the other allele of this gene will also have an inactivation using a copy number deletion. So both alleles of this tumor suppressor gene will be inactivated, so the gene will lose its function, and so this will provide a selective advantage to cancer cells. So to understand all of those mechanisms, so double hits, but also others, we need, and to understand what could be their impact on the cancer progression, we need to have access to sequencing data from which we can look at both at the same time at SNVs and copy number. However, traditional sequencing technologies, such as bulk sequencing technologies, do not have the power to accurately identify all of these events, and only single-cell technologies really has the resolution to identify events happening to very small subpopulation of cells. However, the most common single-cell technologies do not provide an accurate way to look at both those SNVs and copy number at the same time. So indeed, on this side, you can see that single-cell whole genome amplification-based methods offer a quite deep coverage across the genome, which enable an accurate identification of SNVs. However, they compromise on the uniformity of the coverage they provide. And so what I mean by that is simply that if you look at the depth of this coverage, you see that it does not reflect the number of copies that you have for every genomic region. And this simply makes it impossible to accurately identify copy numbers. On the other hand, shallow single-cell whole genome sequencing provide a quite uniform coverage of the genome, as you can see here, to enable an accurate identification of copy number. However, they compromise on the depth of the coverage, and as you can see here, we end up with a very few number of reads per cell, which make impossible to accurately identify all the SNVs this time. So to overcome those limitations, targeted single-cell DNA sequencing came up with another strategy. So instead, on compromising on the depth or on the uniformity of the coverage they provide, they compromised on the fraction of the genome that is sequenced per cell. So here, as illustrated, you can see that we have a pretty deep and uniform coverage, but only for some specific targeted region highlighted here. So for those regions, we can now theoretically look at both SNVs and copy number. However, in practice, this is a little bit more complicated, and actually to call copy number accurately from those data, it is still impossible since there is currently no methods to accurately infer exact copy number. And to understand why this is the case, we actually have to take a closer look to what the data look like. And so here you have an example for which you have the different sequence cells, and for the different genomic positions that were sequenced, you have the observed read count matrix. And if we zoom in to the data that was generated for one single cell, in order to call accurately copy number for this cell, one would need to have data that resemble that. So with very low dispersion per cell, so that reads that come from different regions with different copy numbers, such as 1 and 3 here, end up to be easily distinguishable. However, in practice, the data much more resemble that, where here at the single cell level, you can see that the different copy number distribution overlap and cannot easily be separated. And in fact, there are at least 4 plausible ways to explain the data of these cells in terms of copy number. And I guess we will all have a hard time deciding which one of those 4 options best represent the data of this cell. So because of that, previous methods have simply hypothesized that calling copy number at the single cell level from those data is simply infeasible. However, we're still interested in understanding the interplay between SNVs and copy number. So previous methods, which have the same aim, such as COMPASS, have came up with another strategy, which is to, instead of trying to infer exact copy number, they aim at only inferring a proxy of it, which is only gain and losses. However, if we only try to infer gain and losses instead of exact copy number, we lose a lot of crucial information that would restrain our understanding of tumor evolution when trying to reconstruct the phylogenetic tree. And to give a little bit more, to give a more concrete example of that, here you have a toy example for which you have 4 different clones, and for all of these clones and for the different genomic position, the exact copy number on this side, and the same example where here you have only the gain and losses. And you can see here that if we only look at gain and losses instead of exact copy number, it would be basically impossible for us to distinguish those 2 clones, so the blue and the green one, since they are the same when looking at gain and losses. And this could really confound the reconstruction of the phylogenetic tree of this tumor. So to overcome this limitation, we developed the first methods to infer exact copy number from targeted single-cell DNA sequencing. We called it JIGSAW. And basically, JIGSAW is a Bayesian coordinate descent algorithm that alternates between 2 steps, each solved using stochastic variational inference. This method takes as an input the row sequencing data, so the read count matrix, and output the clonal inference of copy number. So given the input, JIGSAW will first cluster the cells based on their similarity, which will enable the methods to aggregate information across the cell. And this will statistical power to identify the different copy number state inside one clone. Then given this copy number inference at the clone level, it will aggregate information across amplicons this time, so across genomic region, to increase its statistical power to assign each individual cells to its corresponding clone. This 2-step process will be repeated many times until convergence and until we finally get the clonal copy number inference. So to make that a little bit more visual, here you have an example of what the data generated by targeted single-cell DNA sequencing can look like, where you have for every sequenced cell and every genomic region the observed read count. On the right, you will see in a minute the copy number inference made by JIGSAW. On the sides, the 2 color bars represent the true clones and the true copy number clusters. And as you can see here at first, we cannot see any pattern emerging from those data since everything seems more or less mixed up. But here, as you can see, step after steps, JIGSAW will cluster both the rows, so the cells, but also the genomic regions to retrieve the clonal structure of the data. And as you can see, step after step, if you look at the color bars, we see that we get closer to the truth. And for each combination of cell clustering and copy number clustering, JIGSAW will infer the copy number inference until we reach the end, which is this one. So to assess the performance of these methods, we used a previously published simulation tool originally developed for the TracerX study. We extended it to enable the realistic simulation of single-cell sequencing. The reason we used this tool specifically was because we really wanted to replicate as best as possible all the biological, but also the technical challenges that we have identified when trying to go copy number from those data. As such, we can vary the ploidy of cells, the number of clones we want to simulate, the dispersion of the data, as well as the rate of cell unique copy number event. And this enables us to simulate instances with varying level of complexity, as demonstrated by those two examples. So we compared JIGSAW and COMPASS in inferring exact copy number. And as you can see here, we outperform COMPASS in every biological setting tested, and especially when the ploidies of cells increases. But at this point, we were wondering whether this improvement of performance by JIGSAW was only due to the fact that here we are only looking at exact copy number inference when COMPASS was only designed to infer gain and losses. So we compared both methods in their accuracy to retrieve gain and losses instead of exact copy number, and we still found that JIGSAW outperformed COMPASS. And we noticed, to explain that, we noticed that basically JIGSAW has a higher sensitivity while maintaining the same specificity. And this simply means that JIGSAW is able to retrieve more copy number events that were previously missed by COMPASS without increasing its false positive rate. So to understand how and why those improvements could be used to understand some novel biology on the interplay between SNVs and copy number, JIGSAW to 22 AML patients, for which on average 4,000 cells were sequenced using a 37 gene panel, which basically represent a total of 100,000 cells. So the first thing that we have a look at was the frequency of losses across this gene panel. And we found that JIGSAW was inferring more events, and that those events were found to hit key AML tumor suppressor genes, such as NPM1, SMP3, and TP53. And this is not unexpected, since those higher frequencies better match was what was previously found by previous studies made on much larger cohort. We also had a look at the frequency of gain across the same gene panel. And again, here you can see that the difference between JIGSAW and COMPASS is even more striking, where here you have the overall average gain frequency per gene. And this also translates into the fact that those higher frequencies hit key AML oncogene, such as NRAS, KRAS, and MYC. And same observation as before, those higher frequencies inferred by JIGSAW were found to better match what was previously found by previous studies. So at this stage, one question remained, which was whether those new copy number events were overlapping with some SNVs, and if it was the case, what could be the functional effect of that? So to investigate this question, we reconstructed tumor phylogenies for some patient of this cohort. And here I'm showing one case for one patient for which 2,600 cells were sequenced. The copy number inference was made using JIGSAW, and the mutation matrix was inferred using statistical test on the variant reads. And by combining those two signals, we were able to identify 3 different clones, and we reconstructed the phylogeny for those clones. And if we focus on the left branch of this tree, you can see that we identified a double hit event happening on the tumor suppressor genes TP53. So where here the blue clone first acquire mutation on the first allele of TP53, followed later in evolution by this green clone that acquire a loss on the other allele of TP53. And while double hit events were previously reported by previous studies to be indicator of poor prognosis for AML patients, as this event only affect 23% of all the sequence cells, this would have likely been missed by only using bulk sequencing technologies. So to conclude, we introduced JIGSAW, which is the first method to infer exact copy number from targeted single-cell DNA sequencing. JIGSAW was found to outperform previous methods in inferring exact copy number, as well as gain and losses. And when applied to a cohort of AML patients, JIGSAW find more copy number events affecting key AML genes, which reveal new insights into the interplay between SNVs and copy number. So thank you for your attention. I would like to thank the people in my lab, but also my supervisors, without whom this work wouldn't have been possible, as well as my funders and obviously patients and their families.
Thank you. Thank you. Yeah, so we have time for a couple of questions.
Okay. Thank you, Sophie. Nice talk. I didn't understand if you are able to infer exactly also the multiplicity of mutations in these copy number configurations.
So I guess those copy number are not allele specific, but we can extend that and combine the two signals that, because here Jigsaw can only infer copy numbers, copy numbers. But then if we combine that with like the mutation information that we can also get from those data, then it might be possible.
Okay, thank you.
Any other questions? All right, good. Then I get a chance to ask. So I was actually really curious. So you said you have a, it's basically an iterative algorithm that you run till convergence.
Yeah.
Have you found cases, or have you analyzed the algorithm to identify possible cases where you won't see convergence?
I guess the way we implemented it, we do not force convergence, but after a certain amount of iteration, we simply output what we have. But to overcome those probably cases where it does not converge, the way it works is that you try to infer the best copy number inference for a certain number of clones, and you try different number of clones. So maybe it might not work for a certain number of clones, but overall it might work if you extend this number of clones or you reduce it. So it limits that effect.
Okay, yeah. And then following up, then do you see much variation then as you run it towards convergence on different subsets?
Based on the,
like, yeah, do you get the same, do you basically get the same inference of exact copy numbers running it on different subsets?
Of a number of clones?
Yeah.
No, because after we basically select the best number of clones based on model selection. So
All right. Thank you. All right. Thank you. One more time.